Today we have Abhijeet Patil. He's PhD in the Vahidi lab at the University of Pennsylvania, and he's going to be talking to us about a machine learning approach to classify type 1 diabetes in healthy controls using scRNA profiling in pancreatic islets. Do you want to add a little bit to that, Abhijeet?
Yeah, sure. Thanks, Monica. Thanks for inviting for the talk at Sugar Science. So my name is Abhijeet Patil. I did my postdoctoral training under Golna's Vahidi lab at University of Pennsylvania. Our research direction is in understanding the type 1 diabetes using single-cell RNA-seq profiling in pancreatic islets. Previous to my postdoc, I did my PhD from University of Texas at El Paso, and my research focus has been in similar direction in machine learning and computational biology in cancer and diabetes right now. Yeah.
Fantastic, and welcome. We're really looking forward to this talk.
Thank you. So a bit of outline. So I'll go through a quick introduction, objectives, and the machine learning methodology that we developed for classifying type 1D and healthy controls in pancreatic islets, and a conclusion. So as most of you might already be knowing, that the type 1 diabetes is an autoimmune disorder, and it basically happens in like this, as shown in this figure, like 3 stages. The first stage is sort of symptomatic, where you have just less than or equal to 1 autoantibody, and you have normal blood sugar, and the beta cell mass is intact. However, as the disease progresses through time, a person might have more than or equal to 2 autoantibodies, and this causes abnormal blood sugar level, and also the beta cell mass starts depleting. So this is also defined as pre-symptomatic type 1 diabetes, and then ultimately in the stage 3, the beta cell mass is is mostly depleted, and then the person will be declared like symptomatic type 1 diabetes. So basically, the research has always been to focus on early detection of type 1 diabetes. Basically, this might be the window of detection. So there are several major constraints that hampers insight into understanding of early immune signatures of type 1 diabetes. The first major constraint is the inability to safely perform a biopsy of the human pancreas of living donors, and the notable disease progression and beta cell destruction by the time patients are clinically diagnosed. And hence the majority of type 1 diabetes studies have been performed on peripheral leukocytes from the blood, which is not the site of pathogenesis. So to address some of these major constraints, there is this program called HPAP. The HPAP program, which is part of Human Islet Research Network, and is responsible for performing deep phenotyping of human endocrine pancreas to better understand the cellular and molecular events that precede and lead to beta cell loss and dysfunction in type 1 diabetes and type 2 diabetes. So this is part of NIDDK and NIH, and it is supported by several collaborators. Example, Dr. Alinaji is the one is a surgeon who extracts the pancreatic islets, and then the single-cell profiling data is generated by Dr. Klaus Kastner at University of Pennsylvania, and many institutions are involved here. And as we see, there are 3 primary groups involved here. Control, which is right now, as of like March, there are 77 control donors, 30 type 1D, and 33 type 2 diabetes patients. Right? Also, there are a couple of autoantibody positive donors, such as GADA, islet antigen, zinc, so on and so forth. So in total, there are 140 donors as of March. So the objective of our study is to this is the first one is already published. So this study was led by Maria Fasolino at Wahidi Lab and Gregory from from Farabi Lab. So it's single cell multiomic analysis of human pancreatic islets reveals novel cellular states in type 1 diabetes. So here the main so as we see here, there are 3 donor groups, pancreatic islet isolation, and then single cell sequencing. So if you look at the distribution of number of cells across all these 3 groups, there are around 20,000 cells comprising a total of around 70,000 cells. And the cell annotation was performed using a method called too many cells. And as we can see, all the major cellular cell components that are found in pancreatic islets can be seen here. As I mentioned, this data was again generated by Kastner Lab. So the main important discovery here was that MSC, the major genetic susceptibility determinants of type 1 diabetes have been mapped to MSC class 2 genes. This has been known from previous literatures, experiments done through microarray and bulk RNA-seq. So we therefore sought to determine which cell types or donor state disproportionately express genes in this pathway. So here we evaluated the expression of HLA-DPB1, an MHC class 2 gene associated with, known to be associated with type 1 diabetes risk. And the keratin 19 is a marker is a ductal cell marker, which defines the ductal cells. So here we identified that around 11% of the cells among a total 70,000 cells were having the both expression of both HLA-DPB1 and keratin 19. So we looked at the distribution among these cells, which donors they belong to, and strikingly we noted that they were primarily expressed in type 1 diabetes. So this was one of the major contributions from this study, where we found that MSC class 2 is to be type 1. These experiments were also confirmed by several other experiments or assays using IMC, CyTOF, and immunohistochemistry, IHC. So the another major revelation from the study was that this plot, where we have on the x-axis different auto-antibody positive donors, so HPAP29 to HPAP38. So we have around 6 auto-antibody positive donors. And on the x-axis, we have on the y-axis, we have the median's expression count. And the HPAP29 was having low GAD level, whereas HPAP38 was having high GAD level. So there was this direct positive correlation between the GAD level and the expression level, median expression level of these donors. So now that we know a few components such as MSC class 2 and the beta cell expression of median expression correlating with the GAD levels, we continued our research of applying a new approach, basically using machine learning, if we can predict some signatures to identify some early signatures to identify type 1 diabetes, especially within the autoantibody positive donors. So so again, as we saw in Maria Fasolino's paper, we have three groups here: autoantibody positive, where the donors might be having one or two autoantibodies, or more than that in few donors, and type 1 diabetes donors, and then control donors. So we did the tissue isolation, single cell sequencing, and the annotation. So here we have used, we process the data using a single cell transform, which is from Surat, and then annotated using a method called excuse me, called SC sorter, which is a semi-supervised machine learning model. So we have in the data distribution, we have like around more than 100,000 control cells, and around 30,000 autoantibody positives, and around 30,000 type 1 diabetes. So just to validate, if as this is automated method or semi-supervised learning, we just thought of checking if the annotation was right. So we just pulled up the distribution of these donors and across the different cell types. If you see this plot, the type 1 diabetes, as in type 1 diabetes state, the beta cells are should be less. So we saw the same thing in this plot, where beta cells are like low. And yeah, this is the marker feature plot showing the marker expression of PRSS1, which is acinar cells, and glucagon, which is for alpha cells, which is highly enriched in this plot. So yeah, I haven't updated. So this particular data is having I think 67 donors, and it's already published in Nature Metabolism as well. So using this data, we applied the machine learning model. So basically, as some of you might know, machine learning has been used in various fields, such as image recognition, speech recognition, so on and so forth. So the medical diagnosis part, or the medical or biomedical application, is what we are looking into right now. So we have the raw measurements, which is the raw single cell data, and we do the feature engineering, that is to identify the best features, and then apply the traditional model to classify whether it's a disease or a healthy individual, a healthy state. So that's a typical workflow of the machine learning model in here. So this is the complete architecture of our machine learning model. So which includes both gene selection and classification. So we get the raw data, normalize it, which is now like processed, and split the data into 70% and 30% for training and testing. Using the training data, we do the hyperparameter tuning to identify the best parameters across 200 different models, and based on that, we get the best model here ultimately for one iteration. And using that best model, we plug in the test data to predict few metrics, such as a few popularly known metrics, such as accuracy, sensitivity, and specificity. Along with that, as we are using the model, our model is called XGBoost, which is gradient boosting trees. So this model also gives ranked list of genes, which is similar to feature importance in random forest, where you get a list of genes that are used to classify a disease versus healthy state. So we repeat this entire workflow for 100 times to avoid any potential bias, and to build a robust, to get the robust gene list, and also robust results in terms of these accuracy, sensitivity, and specificity metrics. So this particular architecture we applied on all the comparisons that we make, which I'll show in the next slides. So, for example, the first plot, first box plots, we are having all cells, where which means it's unannotated, where all the cells present in the data, for example, in the first plot, type 1 D versus control, all the cells belonging to these two groups were taken together, and we applied the XGBoost model and obtained three metrics. The first one is accuracy, sensitivity, and specificity. So where we see almost more than 95%, more than 95% of accuracies across these three metrics. Similarly, we applied the same thing for type 1D versus autoantibody positive donors and autoantibody positive versus control donors across all cells. So we observed similar trends, although in the autoantibody positive and control donors, we have a decreased accuracies. The reason being autoantibody positive and control donors have mostly similar transcription signatures, and it's hard for the classifier to differentiate these two groups. That's the reason we have a bit less accuracy, but still it's more than, the mean is around more than 90. Next, we applied the same strategy, but now across different cell types for each comparison. So for example, this plot is for all the different cell types within type 1D versus control. We chose all the major cell types such as SNR, alpha, beta, ductal, delta, and immune. And also did the same thing for type 1D versus autoantibody positive versus control. So overall, the classifier accuracy was known to be better compared to better in type 1D versus control and type 1D versus AB compared to autoantibody positive versus control. So our focus in this research is basically on these two groups, type 1D versus control and type 1D versus AB. So here, as I mentioned earlier here, we also, along with these metrics, we also get the ranked list of genes across each of these comparisons. So we obtained first the ranked list of genes across all cells for example, autoantibody positive versus AB versus control, type 1D versus AB, type 1D versus control, and did the pathway analysis using a popular tool called David, and which basically does the which basically uses the Fisher exact test to obtain a significant list of pathways. So we are seeing this KEGG pathways, and some of the pathways of our interest are here, like for example type 1 diabetes and antigen processing and presentation, which is primarily in type 1D versus control and type 1D versus AB. So this was across all cells. Next, we looked into the beta cell comparison of type 1D versus control, and we saw that type 1D and antigen processing and presentation were highly enriched again. So we checked the genes involved in this pathway, and we found that this particular cluster, which we got from a method called ENCODE in Cytoscape, a protein-protein interaction network. So we found these particular genes that were involved in this pathway and also in this pathway. So we checked the expression levels between control group and type 1 diabetes for these HLA or MSC class 1 proteins, and found that most of these or all of these were highly expressed in type 1 diabetes compared to control group. And also the HLA-E is a HLA-1 molecule known to inhibit NK and T cells, and and we see an upregulation of this particular protein in type 1 diabetes patients. So next, so now that we saw HLA class 1 was upregulated in all cell comparison and beta cell comparison, we quickly used, we wanted to predict how the first find where these how these autoantibody positive donors are predicted. So first we use this type 1D versus control as a trained classifier and applied all the donors, all the cells from autoantibody positive donors as a testing set. So we did across all these different cell types. And so yeah, all these on the x-axis are autoantibody positive donors, and so for example, in the first plot, in the SNR plot, on the y-axis, we see that HPAP92, this particular donor had a large number of cells that were predicted as diabetic or type 1 diabetic. So overall, when we aggregate all the autoantibody positive donors, around 10% of single cells from these were classified as type 1D cells. And as we previously saw that in the type 1D versus control classifier, HLA class 1 was HLA class 1 genes were dysregulated or high upregulated in type 1 diabetes. We now compare we just extracted these cells, which are classified as these autoantibody positive cells that are classified as type 1D, and compared along with the other groups. So for example, HLA in beta cell show that misclassified autoantibody positive cells are having similar expression to type 1 diabetes, even HLA B, C, and E. Similar trends were observed even in alpha cells, and also in all cells. In the all cell comparison, we chose these two donors in particular, HPAP92 and 107, because these two donors were the one which were found to be highly misclassified or highly classified as type 1 diabetic in this comparison. So we saw similar transcriptional signatures, like especially for this MSC class 1 for misclassified autoantibodies cells from those particular donors. Next, we applied the same strategy, but now in unannotated cells. So this group shows all the unannotated cells, that is regardless of the cell type. Just use everything we have in the data and plug in the autoantibody positive group as a test set and check where the cells are how the cells are being classified. And we found that HPAP92, again, this particular donor was classified as type 1 diabetic and not control. So on the right-hand side, these bar charts show the selection frequency of the type 1D versus in the type 1D versus control, and all the different classifiers. So which means, as I had shown in the architecture, that we run this experiment for 100 times to improve, to build robust results. So we saw among all those 100 iterations, HLA class 1 proteins mostly were picked all the time, and also few HLA class 2 proteins such as HLA-DRA, HLA-DPB1, all of these were also picked. So we just, as we saw in this plot, that HPAP92 is being having more cells that are classified as diabetic. So we wanted to compare the transcription signatures in this group versus the type 1D for all the MHC class 1 proteins, and we found that the mean expression of these two groups were almost similar or higher in HPAP, this particular donor. So this is all based on the type 1D versus as a training and AB as a testing group. But next we just to cross-check our results, we use this strategy in machine learning called leave one out cross-validation, where so here, for example, we took just to build the confidence of these results, where all the autoantibody positives were classified as like diabetic. So we choose all these autoantibody positive donors, also like type 1 diabetic donors, as a test set in each time. So it is done subject wise. So for example, in one case, all the donors will be used as test case, and one particular donor will be used as a training case, and one donor will be used as a test set. So likewise, we do for all the different cell types, and this is showing the accuracy. So we see that here, all the purple color, this bar indicates the autoantibody positive donors, and HPAP92 is being always predicted as diabetic compared to other groups. So that was one of the major thing we confirmed using a leave one out cross-validation strategy, so which was already observed here. So the overall conclusion is that a major contribution of study was using all autoantibody positive cells, regardless of their cell type, and predicting whether they belong to a healthy or a diabetic state through a trained type 1D versus control classifier built for all cells. So we found that the expression that is up or down of HLA1 or 2 or non-HLA genes between autoantibody positive cells from HPAP92 that was predicted as diabetic transcriptionally matched with the actual type 1 diabetes group. So our ML strategy of applying the powerful combination of gene selection and classification through XGBoost is the first attempt to manifest the higher expression of HLA1 2 genes from autoantibody positive and type 1D donors. Yeah, and also this is the largest single cell data that is available right now, and we applied this. And in future, this work is carried out by postdocs at Vahidi Lab from Mariam and Priya. They are going to apply similar strategies on the multi-omic profiling of immune cells in pancreatic lymphoid. So yeah, I thank all the members of Vahidi Lab, Faryabi Lab, and Dr. Kastner Lab for supporting this work. If you have any questions, sorry, I should have asked earlier, but please.
Yeah, thank you. Thank you very much for this. I see that I think I'm not sure if Miriam's in there in the audience, but I see that Priya is here. And I didn't know if she wanted to join you and kind of just talking sort of high level about the next steps that you inferred in your second to last slide.
Yeah, I don't want to put you on spot, Priya, but if you have any, if you can add anything about like the experiments you're performing, be great.
Feel free to jump in. It's casual. I have a couple of comments. Oh, she can't jump on. We have a couple of comments in our chat, and I'm going to bring those to the group. Michael Kalwat, really cool analysis. Does this approach allow you to identify any circulating markers? This is one of my questions, other than antibodies, that would be predictive in autoantibody plus individuals?
Sorry, can you repeat the question, please?
Sure. You can see it in the chat as well. But it's really cool analysis. Does this approach allow you to identify any circulating markers other than antibodies that would be predictive in AAB plus individuals?
Yeah, I think, sorry, one sec.
Yeah, just asking, you know, can you use what
you're
going to look at any other circulating markers?
Yeah, I think we have the list of markers or genes we identified. Maybe we can check if there is any marker of interest. We can check in our list if it's differentially regulated, basically.
So in terms of sort of the next steps, you know, obviously the billion-dollar question is how to identify some of the endotypal populations for, you know, best treatment or best participation, best individuals to participate in clinical trials. And it seems very interesting what you've got here, that could be definitely applied. How are you thinking about sort of translational steps?
Yeah, I think maybe one of the next step is to correlate the transcriptional signatures with the some of the covariates and yeah, identify if we can predict, if we can have the early detection in terms of combining the covariates along with the covariates. I mean, like the HbA1c or like the different covariates that are used in the analysis. If that answers the question.
Do you have any speculation about the you know, the change that you saw with HLA-E. I mean, that was kind of interesting.
Yeah, that was also yeah, surprising for us. And yeah, it's highly yeah, it's upregulated in type 1 diabetes. And yeah, maybe we will perform some lab experiments and see the difference.
Yeah, that sounds like a really great direction. Also, you know, I guess I'll also ask, what are you pushing this forward in with collaborations outside of Penn?
I think I'm actually like, yeah, yeah, I don't have any idea about that right
Not
now.
sure. Okay.
Yeah, because yeah, I did my postdoc, and yeah, I'm currently working at a pharmaceutical industry since I finished my
Since you
postdoc.
left. Okay, great. Oh, good. Here's one more question. Kevin Harold, can you say anything about inflammatory mediators, if, for example, cytokines or chemokines that are produced by the endocrine cells themselves?
Yeah, we haven't checked those signatures, but yeah, we'll look into those if we have any of those signatures in the list. Yeah, we need to check that.
Yeah, that would be great. A
Thanks.
great piece of evidence to have as, you know, as we're trying, as not we're, but the field is trying to identify who's the best candidate, for instance, for the teplizumab, you know, treatment. Okay, well, thank you so much. This was really great, very interesting, and I can tell a lot of hours went into this work. So really thank you so much for all your contribution, and for the entire GoldNAS lab, the Haiti lab, sorry, and appreciate everything you've contributed and shared today. So we'll keep in touch, and best of luck. We'll be looking for your paper when it comes out. Is it in preprint yet?
Yeah, it's in bioarchive.
In bioarchive. Okay, fantastic. We'll feature it very soon in our weekly newsletter with Weir. Thanks again.
Thanks so much.
Have a great rest of your day.
You too.
Bye-bye.